Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Excel International GCSE Biology Paper 2B for June 2023. Let's begin with the first question. In some areas of the world, the oceans around the coast sometimes glow with a blue light. The photograph shows an area of the coast that is glowing. We can see this is the area. They say this blue light is produced by the presence of thousands of microscopic living organisms called dinoflagellates. These dinoflagellates are protoplasts. The production of light by living organisms is called bioluminescence, a process that has evolved many times in different species of organisms. To generate light, dinoflagellates use special proteins and the ATP produced within their cells. Many species of dinoflagellate contain chlorophyll and are able to photosynthesize. The appearance of glowing dinoflagellates in the sea used to be a rare event, but this now occurs much more frequently. Many of the events occur in the sea around river estuaries, and scientists think that intensive farming and deforestation could be to blame. Due to overpopulation of dinoflagellates in these areas, other species of animal are often harmed. After a series of glowing events, large numbers of dinoflagellates die, causing oxygen levels in the water to decrease. People have often wondered why dinoflagellates glow. They only glow in areas where the water moves around, such as when waves hit a beach. Scientists now think that the production of light is a type of warning to stop predators eating the dinoflagellates. If an animal eats dinoflagellates, the dinoflagellates in the area glow, making the animal obvious to its own predators. To test this, scientists placed dinoflagellates into a tank along with 15 copepods, which are predators of dinoflagellates. When the dinoflagellates glowed, the copepods ate 1,200 dinoflagellates in 2 hours. When the dinoflagellates did not glow, the copepods ate 2,100 dinoflagellates in 2 hours. Some scientists think that we could make use of the dinoflagellates to provide sustainable street lighting. Tanks of dinoflagellates could be placed on top of lampposts. The dinoflagellates would photosynthesize during the day when it is light. A starter powered by a small battery would then move them at night so that they would glow. These sustainable lamps could be carbon neutral and help to reduce pollution. The first part says which of these processes produces ATP in line 6. Of course, ATP is produced through the process of respiration, so the answer should be C. In part B, explain why intensive farming and deforestation would cause an increase in population of dinoflagellates. This is line 9 to 11. If intensive farming is carried out, there is use of fertilizers, and these fertilizers contain nitrates, they contain phosphates, and some of these are going to be organic fertilizers, so these could leach into the water nearby, leading to excess fertilization of the water, causing the increase in the growth of the dinoflagellates. Of course, during deforestation, the trees are going to be cut down, but we know the roots of the trees help to hold the soil together, and if they are cut down and the roots are removed, then the soil is not going to be held together properly, so soil erosion can occur, and all these takes the nutrients into the water, causing excessive growth. So I said intensive farming uses more fertilizers that can leach into the water bodies or dissolve in rainwater towards the sea or towards the water. This provides nitrates and phosphates. These are going to fertilize the water, of course. Also, deforestation removes roots that hold the soil together. This leads to more erosion. And the nutrient-rich water promotes photosynthesis. More proteins are going to be made, and the dinoflagellates are going to grow and increase in number. Next, they say explain why oxygen levels decrease after a series of glowing events. This is line 10 to 13. You remember they said glowing events could occur when an animal or predator has eaten the dinoflagellates. The dead dinoflagellates are going to be decomposed by bacteria, and of course, this decomposition could be aerobic, using up oxygen. Also, due to the increase on these dinoflagellates on the surface of the water, there is going to be less light penetrating through the water, so there will be less photosynthesis of the water plants, and due to that, less oxygen is going to be replaced by photosynthesis. And for this last part, it's because photosynthesis produces oxygen, so if there is less photosynthesis due to less light, there will be no replacement of oxygen in the water, and this is going to decrease. In part C, which type of bacteria converts ammonia into nitrates? This should be nitrifying bacteria because this is a nitrification process, so the answer should be C. Then in part D, they say each of the 15 copepods ate glowing dinoflagellates at a mean rate of 40 dinoflagellates per hour. Calculate the mean rate at which each copepod ate dinoflagellates that were not glowing. We're going to go to line 18 to 21. They said when the dinoflagellates glowed, the corpads ate 1,200 dinoflagellates in 2 hours. And again, remember, there were 15 corpads, so we're going to calculate how many were eaten by each in the specific period of time. Per hour is going to be that divided by 2, giving us this. But these were eaten by 15 corpads, so the dinoflagellate eaten per corpad per hour is going to be 1050 divided by 15, which gives us 70, so the answer should be 70. Then down here they say explain how natural selection could have resulted in the evolution of dinoflagellates that glow. Every time they ask you about natural selection, you have to talk about mutations leading to variation, and then when variation occurs, there is going to be a selective advantage gained. So when selection pressure occurs, those that have the advantage are going to survive, they will reproduce, and then they'll pass on their DNA or their genetic advantage to their offsprings. So here I said mutations could have occurred leading to variation. So the variation is some dinoflagellates gained the ability to glow while others did not. The ones that glowed were not eaten, so they survived and reproduced, and they passed on their allele or their ability to glow to their offsprings. In part A, explain why using dinoflagellates for straight lighting would help to reduce pollution. Because they're photosynthesizing, they're going to be using up carbon dioxide, and this is going to decrease the global carbon dioxide emission. So there'll be less CO2 contributed due to burning fossil fuels, and so many more. So I say this can take up carbon dioxide by photosynthesis to generate the light, so less fossil fuels would have to be used. There would be no net increase in atmospheric carbon dioxide, and they will not produce oxides of nitrogen or sulfur, so there will be no contribution towards acid rain. So this brings us to the end of question 1. Let's continue to question 2. Question 2. A student uses this method to investigate water uptake and water loss by a plant shoot. So they pour 100 centimeters cubed of water into a measuring cylinder and place a plant shoot into the measuring cylinder. They cover the surface of the water with oil, and then they place the measuring cylinder and the plant shoot on a balance and record the total mass. This is the mass before any transpiration occurs. Then they shine light on the plant shoot using a lamp. They record the volume of water in the measuring cylinder. After 4 days, the shining of the light is going to ensure that the stomata are open because the plant will be preparing for photosynthesis, so opening of the stomata will ensure that transpiration can also occur properly. They record the total mass of the measuring cylinder and the plant shoot after 4 days and after 8 days. So this is to see how much water has been transpired. And then the diagram shows the student's apparatus. We can say this is the balance, this is the water, that is the plant shoot. We can see the oil. The oil is going to prevent evaporation of water from the surface here, so we can ensure that water loss is only through transpiration using the plant shoot used. So in part A, they say state the reason for using the oil. Of course, this is to prevent evaporation of water from the measuring cylinder or from the surface, instead of evaporation through the shoot that has been used. In part B, the table shows the students' results. At day 1, we see 100 centimeters cubed of water in the measuring cylinder, and at day 4, we see 75. So the difference is going to be 25 centimeters cubed. Between day 4 and day 8, we see a decrease from 75 to 65, and this is going to be 10 centimeters cubed lost. When we look at day 1 to day 4, the decrease in mass is going to be 175 to 165, which is 10 grams. So 25 centimeters cubed of water were lost, but the decrease in mass is going to be only 10 grams. Here we see from 165 to 155, again the decrease is 10 grams. So between day 1 and day 4, there is more water lost than the decrease in mass. So down here they say the volume of water taken up by the plant shoot is equal to the change in volume of water in the measuring cylinder. This is called water uptake. Calculate in centimeters cubed per day the mean rate of water uptake by the plant shoot during the 8 days. Because they've said during the 8 days and they're looking for the mean rate, I'm going to get the difference in the volume of water, which is 100 minus 65, and that gives us 35 centimeters cubed. So the rate should be the total volume of water lost divided by the total time, which is 8 days. So it should be 4.375, which is approximately 4.38 centimeters cubed per day, if my answer is in 3 significant figures. So that is what I have here. And next they say comment on the changes in the total mass of the measuring cylinder and the plant shoot compared with the changes in volume of water in the measuring cylinder. They say 1 centimeter cubed of water has a total mass of 1 gram. So if we take the density to be 1 gram per centimeter cubed, between day 1 and day 4, 25 centimeters cubed of water was lost, And if the density is 1 gram per centimeter cubed, this is equivalent to 25 grams. However, the decrease in mass is just 10 grams. It means there is 15 grams of water that were lost, and this is because water was taken up for photosynthesis, and not all the water that was lost from the measuring cylinder was transpired to the air. However, after day 4, we can see the 10 centimeters cubed, if this corresponds to 10 grams, and the total mass lost is also 10 grams. It tells us the mass of water lost from the measuring cylinder is equivalent to the mass lost from the total plant. So here I say there was a decrease in both the total volume of water and the total mass. For the first 4 days, there was greater decrease in volume, 25 centimeters cubed, which is 25 grams, than in mass, which was just 10 grams. For 4 days, the volume of water taken up by the plant was equal to the volume of water lost, meaning the decrease in total mass. So through transpiration, water was taken up by the plant for photosynthesis, sugar, and transportation of minerals. Next part says, explain why the rate of water loss would be different if a working fan is placed in front of the plant shoot. If a working fan is placed, it's going to decrease the humidity in the air around the plant shoot, and that is going to cause more transpiration because there will be a greater diffusion gradient. The rate of water loss would be higher because more transpiration would occur. The wind from the fan decreases the humidity around the plant, so a greater concentration gradient will be created for faster rate of water loss. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the diagram shows part of a human nephron, which is what we see here. The first part says which structure is a proximal convoluted tubule. A is going to be the proximal convoluted tubule. This is the distal convoluted tubule. We see the loop of Henle, the collecting duct, and so on. So the answer here should be A. In part B, the hollow space inside a blood vessel is called a lumen. The blood vessel labeled X has a lumen with a radius of 100 micrometer. Use this formula to calculate the cross-section area in millimeters squared of the lumen of this blood vessel. So they want us to use the formula area of a circle is equal to pi r squared. So from here, I know this is my radius, which is 100 micrometer, but they've given me that 1 millimeter is equal to 1,000 micrometer. If 1,000 micrometer equal 1 millimeter, then it means 100 micrometer are going to equal to 0.1 millimeters. So this is my radius. So the area should be pi times radius squared, which is pi times 0.1 squared. And if I use pi as 3.14, this is going to be 3.14 times 0.1 squared, which is 0.0314 millimeters squared, to 3 significant figures, and this is going to be my answer. Here they say the lumen of blood vessel X is wider than the lumen of blood vessel Y. Explain why this difference in the width of the lumen of the two blood vessels is important for kidney function. If we go back here, there is blood vessel X and blood vessel Y. This is smaller in comparison to this, so that pressure generated here is going to be greater for ultrafiltration to occur efficiently. So I said blood vessel X is wider than blood vessel Y, and this leads to increased blood pressure. So ultrafiltration can occur, so the plasma and other smaller substances enter the Bowman's capsule. This is to ensure that ultrafiltration occurs efficiently. In part C, protein is often found in the urine of people who have high blood pressure. Describe how urine could be tested for protein. Here we use the biuret test. Of course, this biuret test contains copper sulfate, so it's going to be blue initially. However, it will turn purple due to presence of proteins. So this brings us to the end of question 3. Let's continue to question 4. Question 4, the diagram shows a cross-section through part of a plant leaf, which is that we see the palisade cells, the spongy cells, the guard cells, and so on. So they say, what is the name of the cell labeled P? P is a guard cell because across two guard cells, that's where we have the stomata. So the answer is going to be B, guard cell. Next, they say, explain how part Q is adapted for photosynthesis in the leaf. Part Q contains the spongy cells as well as the xylem and phloem. So we can see here the xylem and phloem, and then the spongy cells. The spongy cells contain spaces between them, so that carbon dioxide can pass through them towards the palisade, where there is going to be more photosynthesis taking place, and the produced oxygen can live through them towards areas where it's going to go. Also, we have the xylem vessels that are going to deliver water, which is a reagent required for photosynthesis. So I said part Q contains the spongy cells. The spongy cells have air spaces between them, So efficient diffusion of carbon dioxide occurs towards the palisade cells for photosynthesis to occur. And again, remember, photosynthesis can also occur in the spongy cells, although greater photosynthesis occurs in the palisade cells. Also, part Q contains xylem vessels to transport water for photosynthesis. In part B, a student uses this method to investigate the effect of temperature on the rate of gas exchange in leaves. There is a pour 10 centimeters cubed of hydrogen carbonate indicator into each of 6 test tubes. Hang a leaf in 5 of the test tubes. Place a bang in each test tube, and then place the 5 tubes with leaves into separate water baths at temperatures of 15, 20, 25, 30, 35, and 40. Place the tube with no leaf in a water bath at 25. This is going to act as a control so that we can know any changes that occurred are due to the presence of a leaf and not due to any other condition. Then place all tubes in bright light. Record the time taken for each of the hydrogen carbonate indicator solution to change from orange to red. The student repeats the experiment 2 more times, and the diagram shows one of the tubes with a leaf. So we have a bang, we have the leaf, and the hydrogen carbonate indicator. This helps us to test for presence of carbon dioxide, depending on which color it changes to. So they say state the independent variable. The independent variable is the thing you change during the experiment, and because here we're changing temperature, we see temperature is going to be the independent variable, or you could say the temperature of the water bath. The next they say the species and size of leaf were the same in each tube. Give a reason for controlling one other named factor. The other named factors, of course, you can say pH, carbon dioxide concentration, light intensity, and so on. So I chose change in pH. This should be enzyme substrate complex. Change in pH affects the active sites of the enzymes, and this affects the rate of reaction because it's going to affect the rate of formation of the enzyme substrate complex. Remember, if the shape of the active site is changed, then this complex is not going to be formed, and so the rate is going to be affected. Next, they say set the function of the tube with no leaf. This is going to be a control experiment. Like I already said, this is going to help us show that the observed changes are due to presence of a leaf and nothing else. And in part C, the table shows the students' results. We have the variation in temperature from 15 to 35, and the time taken for the indicator in tubes with leaves to change from orange to red in minutes. So we can see there is 50 to 15. As the temperature increases, the time taken for the color of the indicator to change from orange to red decreases. I'm going to take the mean because the mean is more representative. From 43 to 37 all the way towards 12, we see from about 30 degrees, the time taken does not change or does not decrease further. So it's going to be constant, and that will tell us that temperature is no longer affecting the rate of photosynthesis, but some other factor is affecting the rate. Calculate the mean time taken for the indicator to change from orange to red at 25 degrees Celsius. I want you to give your answer to 2 significant figures. We go back to the table. At 25, I'm going to take that, and that, divided by 3. So I'm adding 25 plus 30 plus 25 divided by 3, and I'll get the answer for this point here. You can see I got 27, and you could write 27 in this space here. Explain the effect of increasing the temperature on the mean time taken for the indicator to change from orange to red. Of course, the higher the temperature, the shorter the mean time taken for the color to change, like I've already explained from the table. So as the temperature increases, more carbon dioxide is going to be removed from the tube because more collisions occur and the rate of photosynthesis increases. And again, remember, with more photosynthesis, there is going to be more carbon dioxide used up, so the color of the indicator is going to turn from orange to red faster if there is a faster rate of photosynthesis. However, after 30 degrees Celsius, the mean time taken is constant because another factor other than temperature limits the rate of photosynthesis. Then in part D, in another experiment, the student places a test tube containing a leaf and hydrogen carbonate indicator in a 25 degrees Celsius water bath. The student then places all these apparatus in the dark for 1 hour. Explain why the indicator solution changes from orange to yellow. If you place this apparatus in the dark, there is going to be less photosynthesis, so more respiration will take place, and respiration produces carbon dioxide, so the concentration of carbon dioxide is going to increase, and that's why the indicator changes from orange to yellow. So I said in the dark, respiration occurs at a faster rate than photosynthesis, so more carbon dioxide is produced and the indicator turns to yellow. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Hormones control the menstrual cycle. Name the gland that produces follicle-stimulating hormone. This should be the pituitary gland. And next they say the diagram shows the changes in 4 hormones during the human menstrual cycle. Of course, this is going to be estrogen that increases because it's nourishing the walls of the uterus or the uterine walls. We see FSH increases, and then we see luteinizing hormone to increase. FSH increases to stimulate the egg to mature in the follicles, and LH increases to cause the egg to be released from the follicles, or you would say ovulation to occur, and progesterone increases after ovulation to maintain the uterine walls. So the first question says, which line represents the hormone progesterone? Of course, it's going to be this one here, so it should be D. Then they say describe the role of FSH and LH in the menstrual cycle. Like I've already said, FSH causes the eggs to mature inside the follicles. It also causes the release of estrogen. Luteinizing hormone, it stimulates the release of estrogen or progesterone, and it causes egg release, which is ovulation. In part B, they say reproductive hormones can be used as contraceptives to prevent pregnancy. The table gives information about 3 different methods of hormonal contraception. There is oral tablets. Tablets can be taken every day at the same time. So here we see every day, meaning there is a chance that somebody could forget. So here the percentage effectiveness in preventing pregnancy is just 91 to 99. And if compared to the others, you can see there is less effectiveness here in comparison to the others. There is injection into the muscle by a medical professional. So here we need to visit a doctor's office, and it's going to be painful because it's an injection. And then injection is repeated every 12 weeks. So because it's repeated every 12 weeks, there is a chance that you will not forget, and it's going to be kind of more effective. If you compare 94 to 99, it's more effective than tablets. When we go to implants, a plastic rod containing hormones is surgically placed under the skin of the upper arm. This could be painful, and you have to seek a professional's help because it's surgery. They say it can last up to 3 years, and then it needs replacing. So because it takes longer periods of time, it's kind of convenient. However, there is need to replace it, meaning you have to go through surgery again. So it's going to be inconveniencing in the long run. However, you have 94 to 99% effectiveness, which is good. These 2 methods involve seeing a medical professional, but here you can just take tablets. However, there is no surgery in this method. So all methods have advantages as well as disadvantages. Then they say discuss the advantages and disadvantages of the 3 methods of contraception shown in the table. So I'm just going to read what I wrote because I've already gone through the point. Overall, the oral tablet has less effectiveness than injections and implants. Oral tablets have 91 to 99%, while the injections and implants have 94 to 99%. Oral tablets need no medical procedure or surgery, so there is no pain. However, they have to be taken every day and at the same time, and this requires more dedication. Injections are only given every 12 weeks, and there is no need for daily intake, so it is hard to forget, like with oral tablets. However, medical professional is required, so a visit to the doctor's office may be required, and that could be kind of inconvenient. Implants are long-lasting, period of 3 years. However, they must be surgically inserted, so there is pain, and the chance of uncontrolled hormonal release could occur, so they also need to be replaced. This brings us to the end of question 5. Let's continue to question 6. Question 6, the photograph shows a plant called a snapdragon, which is this one here. They say selective breeding has been used to produce snapdragons with brightly colored flowers. In part A, describe how selective breeding can produce snapdragon plants with brightly colored flowers. When you are using selective breeding, it means we have to choose plants that already have bright colored flowers, and then we're going to cross them. Then we'll produce offspring and choose among the offspring plants with the brightest flowers, and then we cross those over generations. So I said select snapdragon plants with bright flowers and cross them. Then cross the offsprings with bright flowers. Repeat crosses between offsprings with bright flowers for generations. Micropropagation is often used to make copies of a snapdragon plant. Describe the process of micropropagation. In micropropagation, we can take small tissues or cells from the snapdragon plant. We're going to disinfect them, The disinfection is to ensure that microorganisms are not taken along with the actual plant tissues to be cultured on the nutrient solution, because they will compete with the plant tissues for nutrients, and that will not be good, or they could even cause infection. So we disinfect the sample to remove microorganisms. This could be done using ethanol. Then we place the plant tissues on agar plates with correct nutrients. The agar plates will contain the correct medium to ensure that the plant tissues can actually grow into actual plants. Then we add root and shoot growth stimulating substances like auxins to ensure that actual plants can be formed, which can later be planted into the soil to grow as actual plants. In Part B, scientists investigated the effect of exposing snapdragon explants to increasing amounts of ionizing radiation. This is the scientist's method. Take a snapdragon plant and use micropropagation to produce many explants. We're producing many explants using micropropagation because they're going to have the same DNA. Since we are testing the effect of ionizing radiation, then they say expose groups of explants to different amounts of ionizing radiation, then grow the explants into plants and record the number of differences in their phenotype compared with the original plant. Then take samples of each of the plants and measure the number of differences in the DNA nucleotides of each plant compared with the original plant. Give the reason why micropropagation is used to produce the plants to be tested. Because we are testing the effect of ionizing radiation, we have to ensure that the plants we begin with have the same DNA or are genetically identical. So X plants have the same DNA, so I said, so that all the plants used are genetically identical. This ensures that any differences we see in the phenotype is not due to the differences in their DNA, but is going to be due to the effect of ionizing radiation. The next they say what term is given to all the DNA in an organism. This is the genome. So the graph shows the scientist's results. We have the number of changes in DNA, which are these, and the number of different phenotypes. From the graph we see there are more genetic changes than the changes in the phenotype. And this could be some changes in the DNA are not going to be transcribed into messenger RNA, which can later be made into proteins. Remember, the proteins produced are the ones that change the phenotype. So if some of the mutations that occur in the DNA are not transcribed or converted into proteins, it means there are fewer changes in the phenotype because some changes in DNA were actually not changed into proteins. Discuss the effects that increasing the amount of ionizing radiation has on the snapdragons. Also, of course, the greater the ionizing radiation, the greater the number of changes in DNA as well as the number of changes in the phenotype. This is because ionizing radiation leads to mutation in DNA, of course. These are going to be changes in the DNA. There are more changes in DNA than the changes in the phenotype because some mutations are silent, meaning these mutations occur in sections of DNA that are not going to be converted into proteins. Some may not lead to changes in the proteins produced. Other mutations may occur in regions of DNA that are not used to make proteins. So this brings us to the end of question 6 as well as to the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.